University Ibadan here in Nigeria, and I'm also an associate of the Medical Laboratory Science Council of Nigeria. I think I'll keep it at that. Of course, during the course of this training, we will be going through metagenomics. That will be our focus through the next 7 or 8 days of this program. But then I'm sure many of us are quite conversant already with our parent company, but then I will still, by way of giving acknowledgement to where it is due, talk about them, and of course, for some persons who are joining us for the first time on a day like this, so you get to know about Omics Logic and the Pine Biotech Incorporated USA. But to start with, um, this is what we will be engaging ourselves with in the next um, couple of days. Omics Logic Metagenomics Workshop Part 1. For some of us who might have seen our flyers, we might already be aware that there's a Part 2 to this that will be coming up almost immediately after the end of this program. That will be 30th of September to October 7th, thereabout. I'm sure we'll speak more to that afterwards. All right, but it is my pleasure to welcome every one of us to this Metagenomics Workshop Part 1. Of course, the program is designed to highlight the metagenomic specialization and to help each and every one of us learn to explore how different types of analysis can be used to understand the microbial world, and of course show us different bioinformatics approaches for this analysis of the microbiomes. My hope is that we'll get the very best from these information sciences and tools that would be shared during the course of this program, and subsequently, of course, we will not hesitate to equally share with us some other tools or platforms or extraneous, I mean beyond even the learn platform, that can help us in our researches or in our work. However, most of what we'll be learning in this course will form the background and basis to gaining more or doing more with metagenomics. But as I did mention earlier on, Omis Logic has a goal, which is to provide training in bioinformatics, enabling independent research guided by mentors and peer examples. Okay, this is a critical part of what we are trying to do, to provide this training to individuals so much that yourself, every participant can begin to see how they can conduct their own independent research, following, peer examples, projects, and other, as well as guidance from mentors, to ultimately get something done. So that is one of our primary goals. And of course, we want to also develop a community that has a shared passion for data-driven research and appreciation of citizen science. And of course, we would be enabling students, clinicians, and faculties of all backgrounds to also develop these novel and independent research, frameworks using latest technology in life and data sciences. I would like to, acknowledge the team behind the Omics Logic programs, Pine Biotech, and Tauber Bioinformatics Research Center, starting from the head of the team, that is Dr. Fred Tauber, whom we are, heavily leveraging series of works done over the years with a whole lot of impact in oncology, immunology, biochemistry of inflammation, and the rest, as well as Dr. Leonid Brodsky, who has done a lot of research and works that has produced a lot of bioinformatics algorithms, some of which are out there and many of which are part of this platform, as well as Mr. Eliabrowski, Dr. Mohit Magzunda, who is the project and international lead, and several others time will fail me to mention. Equally here on the Omis Logic African team, these are the team working together to bring programs across participants from Africa and beyond, and equally we are doing our best, and I can tell you, I mean, the team is also growing. Maybe next time during our next program, this slide might have changed. All right, so moving on, the Omics Logic Metagenomics workshop. We are currently um, undergoing the opening live section, where we just introduce us to what is to come and provide some basic information or background to what is to come during the workshop series. Let me mention that this program is a blended program, or it's happening virtually, and it's a blended one, in the sense that participants are expected to go through, I mean, to participate or by themselves take on the self-paced courses on our platform that we will be giving access to those who are fully registered. And of course, the WhatsApp platform will serve to provide the forum through which we answer questions and take on any concerns that individuals might have. All right, so that is what we will be doing. Course one is Introduction to Metagenomics, or simply put, Metagenomics. It has 13 lessons, and I bet it will be providing us a lot of background information to what we will be doing, and how to utilize the metagenomics frameworks to undergo or conduct a research. Course two is on role of high fat diet on obesity. I can tell you this is a project oriented course. All right. It is actually a project. So going through that project, or through that course, I want to believe it begins to elicit, the mindset of running your own project in individual participants. You see the tools and the steps they take to run that, to conduct that complete project. All right, it's a self-paced one equally. And of course, course three is effect of dietary fiber intake on microbiome. I must say this is going to be a week of immersive learning. It's going to be a week of immersive learning. Course one, I think, averagely, on the platform, it's a course that should take 25 hours. 25 hours is a day and 1 hour. All right. But of course, we do not expect that, you do that at a stretch. Yes. So you're going to be engaging those lessons and taking those, completing them as the course and the days progress. All right. But course two, especially role of high fat diet on obesity, it has 7 lessons, but it will surprise you that the estimated time is 50 hours. All right, meaning you are going to engage so much of, the content, running a whole lot of, tasks and assignments within that course. And of course, course three equally has 25 hours estimated. So please, I want to encourage, for those of us that will be taking these courses, having been access activated to them, please hit the ground running and start taking them. Don't procrastinate, please. I mean, build successively on your pace. I'm sure you will have several other things to equally still do, and I can bet you can actually complete these courses within these 8 days. 8 days is about 8 times 24 hours. So I want to believe we can complete all of these, courses. Of course, we try to see how to have a closing live session, one more live session like this, to discuss how to move on from there. So, on that note, I welcome us to this bioinformatics training with focus on metagenomics, and I will be handing over shortly to my partner, who will take us through, the next set of slides on session, before perhaps I come up again towards the tail end of this program. So, on that note, I welcome every one of us, and I will want to introduce to you now Dr. Ayofolo Alabetutu, who will be taking the next session. Over to you, sir. Are you there?
Yes, I am here. Thanks so much for that brilliant introduction. It's so lovely to have every person's join us here, looking at all our research interests. It's quite lovely to see how that our research interests uh, differs. So on that note, I would love to share my screen. Please do confirm that you have seen my screen. I believe we can see my
Yes,
screen.
I think yes, I do.
All right. So real quick, I'm going to introduce myself. My name is Ayonfiolua Alabetutu. I am a Nigerian and I'm an African. I am a molecular biology and genetics expert from Penn State University. I am a researcher, bioinformatician, uh, who works with the Federal Research Center of Translational Medicine in Siberia and Russia. I double as the community engagement manager with Omics Logic Africa and UREKA here in Nigeria and for Africa. And I will be your host for opening the session of this workshop. So please stay tuned and let me know when I am going so far. I hope you follow me to this point. Do you?
Yes, I do.
Please.
Okay. All right. All right. It's good to see some of these feedbacks. Yeah, it's good to see some of these feedbacks. All right. So on this note, I would love to just take us on the journey of what we have before us. Metagenomics. In this opening section, we'll be discussing on around some interesting things. We'll be having an introduction speaking to metagenomics. We will go further to talk about genomics, proteomics, and some of the omics um, that all focus on the whole of a single organism, and we'll touch the organism's life in its community and how the organism reacts. We will also try to examine how to study a community, looking across the genome of the organisms living in it. Uh, this is actually the focus of metagenomics. So we will, as much as possible, try to understand what metagenomics is, and also strive to also understand what metagenomics allows and what metagenomics does not allow. I believe that I am trying not to go so fast so that we can all follow me, and let's get started. So metagenomics is a study of metagenomes, genetic materials sampled directly from an environment. So it allows the study of communities of organisms directly in their natural environment. And so it means that this kind of study helps us to bypass the need to isolate and the need for lab cultivation. So metagenomics, it refers to both the research techniques and the research field, the word metagenomics. So metagenomics as a field can be defined as the um, genetic analysis of microbial DNA from environmental community. Then metagenomics tools actually enables the population analysis of unculturable or previously unknown microbes. So it is important to take note that only 1 to 2% of bacteria can be cultured in the laboratory. I want us to take note of that. The ability to identify microbes without a prior knowledge of what a sample contains is opening new door in the discipline of microbial ecology, in virology, in microbiology, in environmental science, and biomedical research. Sequencing based examination of the metagenome has become a powerful tool for generating novel hypotheses. So what we can see on the screen, the omics technologies for microbiome studies, we can see two interesting ones here, right? We can see the DNA-based approach, and we can see the RNA-based approach. And we can also see another approach over there, but we will try as much as possible to stick with the DNA and RNA-based approaches, right? I don't think we're not going to touch metatranscriptomics here, but we will try as much as possible to stick with 16 Hz, 18 Hz, ITAs genes sequencing. Yeah, but we can also see that there are all other um, approaches to study microbiome, and except from the DNA based approach and the RNA based approach, we also have metaproteomics, metabolomics, and this is quite interesting because one actually focuses on the interaction with protein, and here we're just going to study what proteins is being used, and metabolomics kind of also focuses on what chemical outcome of activities are actually observed. So where metagenomics, metatranscriptomics, where they kind of focus on sequencing, metaproteomics, metabolomics also focus on sequencing to an extent, but at the same time there is an interplay with other with another concept, and the concept is known as mass spectroscopy. So not to belabor us with some of these big matters, let's just come straight away and let us try to limit it again. Let us try to limit what we're going to study today, or the outlook we rather have to the DNA-based approach. Yeah. So what do they entail? What can they do? We'll try to focus as much as possible. We will try to focus on 16S RNA, 18S ITS gene sequencing and code, so that we can get to understand that one first, then we can probe further if we're kind of interested in others. So today we know that the endogenous human microbial community contributes to critical metabolic, physiological, and immunological functions, and they include differentiation and maturation of the host mucosa, its immune system. They also interplay within food digestions and nutrition. Definitely, we know that microorganisms play quite a strong role in energy generation and conservation. It also play a great role in metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals. I'm just talking generally about. I'm still talking about the microbiome function. It plays a great role in maintenance of your skin and mucosa barrier function. It plays a great role in the development and regulation of the immune system and fine-tuning of its reaction pattern. That is the balance between pro-inflammatory and anti-inflammatory processes. It plays a great role in the invasion and the growth of disease-promoting microorganisms, colonization, resistance, and so on. I'm speaking all of these things to speak to the fact that some persons don't actually know what area can they have their own research in metagenomics. So, are you looking for an idea of what project you might be interested in? There are interesting concepts here. You can study the immune system with it. You can study food digestion, nutrition. You can study energy generation. You can study all of what you can see here on the board, even onto its functional level. So I remember that somebody in this training at one point we had something known as peak rust, so functional level, right? So we are going to get there very, very soon. So let's just go back to the basics, so that because the basics never grow old. There are 3 microbiome sequencing types. We can see it on the screen. The whole genome sequencing. We have the 16S RNA sequencing. We have the amplicon. I shall throw light on this. The whole genome sequencing, also known as the complete genome sequencing or the entire genome sequencing. It's a process of determining the entire or nearly the entirety of the DNA sequence of an organism's genome at a single time, right? At a time. And this entails sequencing all of an organism's chromosomal DNA as well as the organism, as well as the DNA contained in the mitochondria. And for plants, for example, you have to focus on the chloroplast, right? The chloroplast DNA contained in the chloroplast. So 16S RNA is a type of sequencing. 16S rRNA sequencing is a type of sequencing with the R standing for ribosomal. So 16S rRNA means 16S ribosomal RNA sequencing. So in this kind of sequencing, the RNA component of the 30S small subunit of the prokaryotic ribosome, that's the SSU RNA, that is what we try to focus on. So it binds on the Shine Dalgano sequence, and it produces most of the small subunit SSU structure. So as much as possible, understanding of how the ribosome work and how subunits of the ribosome are and how they binds are actually what 16S ribosomal RNA sequencing pivots on. And this is actually one of the greatest tools that you will want to work with in your metagenomics journey. Then we also have the amplicon. An amplicon is a piece of DNA or RNA. I'm talking about amplicon sequencing here. So an amplicon is a piece of DNA or RNA that is the source or sometimes is the product of amplification or replication event. So it can be formed artificially. Amplicons can be formed artificially using various methods, including PCR, LCR, that's polymerase chain reactions, ligase chain reactions, or naturally through gene duplication. So that is that about that. I want you to start writing down your questions because I understand that I have started approaching some uncharted waters. I believe so, right? Please, if you follow me to this point, I want you to just go to the chat and say I follow real quick, and I shall continue. So before going straight or deep into the techniques, let's understand what is the ribosome, right? Can any one of you tell me what is a ribosome? Someone just unmute and tell me what crosses your mind. Let's just have a quick rehearsal or a quick revision. What is a ribosome? What crosses your mind when you hear the word ribosome? Can I see someone's hand?
Yes. When I hear the word ribosome, what comes to my mind is that ribosome is the site of protein synthesis.
Oh, great. Do I have any other thoughts again? Okay, site of protein synthesis. Do you have any thoughts again? All right, thank you so much for that submission. I believe others are following, but there's just wondering that, okay, I think she has said what I want to say. So the ribosome, of course, is at the core of translational of the translation machinery of all organisms. So and it assures, I mean, we know it for two core functions. The decoding of the genetic information contained in in the messenger's RNA, and the formation of peptide bond. So ribosomal rRNA, they are actually the main structural and the catalytic component of the ribosome. So ribosome, I would love to refer to it as a molecular machine that translates RNA into protein, right? And so it is, I mean, looking at this molecular machine, we need to understand that there is actually a rate of change of certain regions of the ribosome of the ribosomal gene is something that makes us kind of interested in studying the rRNA. So, um, it makes us understand that so the way the ribosome actually work uh, is kind of interesting. Before I go to talk about, before I go deeper to talk about the difference between prokaryotic and eukaryotic ribosome, I need to also to say this again. This molecular machine called the ribosome, right? It has certain regions in it. And this region, I'm talking about the regions of the ribosomal genes. And these regions make us interested in studying it. Why and how? Because the ribosomal genes, because certain regions of the ribosomal genes make this molecular machine a contemporary standard for life detection. So there are some things going on in this molecular machine called the ribosome that we want to get to know, and that after a while, after they have studied it, yeah, it became a target for, to be used as a point for sequencing. Even to the point that we use, it became, we had a 16S ribosomal RNA sequencing. It became a target. So real quick, here are some differences between the ribosomes of prokaryotes and the ribosomes of eukaryotes. Yeah, we can see that the prokaryotes, they have few ribosomes, and whereas the eukaryotes have large ribosomes that facilitate translation in eukaryotes. The ribosomes in prokaryotes are found inside the bacteria and the archaebacteria. Yes, we can also find ribosomes in the eukaryotes. You can find them in animals, plants, fungi, and other unicellular eukaryotes with a nucleus. A quick question. I want someone to go. I believe that a lot of us are microbiologists, and some others are zoologists, and some others are researchers, clinicians. I want you to go quickly into the chat and write an example of a unilateral eukaryote with a nucleus. I want you to go to the chat and write an example of unilateral eukaryote with a nucleus. So we can also see here some differences. One of them actually has to do with the molecular mass, the small mass. We can see that relatively for the prokaryote, the mass of its ribosome differs, and for the eukaryote, the mass of the ribosome actually differs. Then we can also see that the sedimentation coefficient also differs based on its weight. Then we can see some other details here that will be quite interesting for you to go pursue and try to read up. So real quick, I want to still touch more on the ribosomal RNA. Let me go to the chart to see who actually answered some of those questions. So I asked that I wanted to see an example of I asked for an example of a unilateral eukaryote. Give me an example of unilateral eukaryote with a nucleus. An example of unilateral eukaryote with a nucleus. So that was the question. Okay. As we try to answer that question, let's just continue. So as the ribosome is universal and submitted to strong selection pressure, ribosomal RNA has been extensively used as a reference molecule in phylogeny and at the origin of the division of living organisms. So we have been able to use the ribosomal RNA as the tool or the object to divide living organisms into different domains. So from the bacteria to the eukarya and to the archaebacteria. with the ribosomal RNA, we can separate them. So we can actually see some here. The ribosomal RNA gene, this gene have been known for many years to vary much less across the tree of life. So across the domains, the phylums, they actually differ from one another. So it's actually a ribonucleoprotein particle, and this particle is actually measured in S, like a lot of us have actually edit over and over again. So we find that 17S is 70S in prokaryote and 80S in eukaryote, and it's composed of 2 subunits, right? Please, I want us to understand that the subunit summation is not mathematical. So because someone be wondering that if the prokaryote total ribosome is 70S, sorry, I'm talking about the large and the small subunit. How is it that they're able to add 50 and 30 and they're getting 70? So I want us to know that this summation is not a mathematical summation as it were. So deeper work into how the ribosomal RNA works, as we examine its subunit, we see a variation. We see variations in its composition. 16. I'm trying as much as possible to make it not too complex, yeah, as it were. So the gene coding for the for the ribosomal RNA actually helps us to be able to separate, to understand how organisms actually differ from one another. So I need us to understand that from species to species, even onto the species level. Okay, let me just come up a little higher. From the family level. So the more you come down the tree of life, from the kingdom to the phylum to the order, and you keep on coming down, you get to understand that no 2 species has the same 16S rRNA. So this has actually become a tool because the subunit weighs differently because of the number of proteins it's able to produce, and in the long run, the approximate number of proteins, that's the S that can be produced in each of the subunit of maybe prokaryote, maybe eukaryote, helps us to be able to use it as a means to categorize that, oh yes, I have caught this species. Why? Because it has this number of, it's able to produce this number of protein, and I'm going to explain how we reverse play this into reconstructing phylogenes. So if I'm able to understand that, okay, in a particular kingdom, in a particular phylum, taking it all down to a species levels, if I'm able to trace it all down, that okay, this is the number of protein this one can produce, this number of protein this one can produce, I'm able to practically trace down or start to make phylogenetic constructions already. So due to the slow rate of the evolution of this region of the gene, so this region of the gene to an extent is stable, and we are able to use it for this purpose. So 16S rRNA is a taxonomic genomic marker. It is common to almost all bacterial and archaebacteria. So this marker actually allows us to examine genetic diversity in microbial communities, specifically specifically to help us to know what microbes are present in the sample. I am trying as much as possible to to make sure that it makes sense to an extent. All right. I believe that we have been able to. I mean, we actually followed until this point. Okay. At this point, I will go further to speak to some of the other concepts that actually that we need to take note of, because I understand and I know that a lot of us here we are all about the wet lab, we are all about some of these other details. I hope we are able to bring clarity to that as soon as possible. So let us go back to what we have here. Metagenomics, again, is a study of the total DNA extracted from an environment at a point in time. So in this case, let us try to use a human body sample, for example. So also, I mean, we can note that, okay, this environment does not, can also be the soil, water body, you can do wastewater sequencing, animals, you can collect samples from the gut, any environment, okay? So, but I'm going to set an example now. So let me set an example that I'm collecting a sample from an environment, and the environment is the human body. So instead of having to culture these specific microorganisms, right? What I'm going to do is that I'm going to extract its DNA. And then what do I do after extracting is DNA? I try to charter it. I mean, to shatter it and then sequence it, right? So this is what I need to do. Practically, this everything you need to practically know about the sample collection to the sequencing. Now, I want you to take note that there are lab works you need to know about extracting of DNA, about shattering it and amplifying it. So please just take note. And collection, how do you collect it? Where do you put it and co? Please, these are other details that we don't need to take for granted. But let me come back to metagenomics. So metagenomic sequencing actually allows scientists to directly extract and sequence DNA from their environmental sample. So one of such method for this sequencing is a method we called the shotgun sequencing. So I remember that Angie wrote that she's interested in whole genome sequencing. I think maybe she also wrote that she's interested in shotgun sequencing. So in order to sequence the metagenome of the human microbiome, shotgun sequencing is actually one of the methods. So what does shotgun sequencing, what comes to mind when we talk about shotgun sequencing? We're going to get to understand that concept in a bit. But note, in shotgun sequencing, all the pieces of the DNA within the samples, they are cut into short pieces. Then after the DNA is cut into short pieces, then we try as much as possible to sequence it. So that's a method of shotgun sequencing. So it's going to many a times produce short reads. Okay. At this point, I would move to metagenomic sequencing. What comes to mind when we talk about metagenomics sequencing? What can we see? How can we see it? What do we need to understand? What is even obtainable from metagenomic sequencing? So I'm going to try as much as possible to run this again so that our eyes can follow this flow chart that we have on the screen. We have the microbes to the sampling, to DNA extraction, library construction. We can see the sequencing machine. We can see the sequencing read. From the read, there are a lot of things we can actually see here. And tada, you can start to see what taxonomic profiling, gene and functional profiling, and it continues. with the taxonomic tree and co, you can do a lot of things. You can start to study abundance. You can start to study evenness. You can start to study the quantity, the balancing. I mean, there is something known as having good reads to know if you've collected enough samples and co. Alpha diversity. Then you can start to do some other beta diversity, intra sample diversity. That means that's a contrast of what is present between two samples or between two or more samples, and start to draw and do your other analysis, PCA, non-dimensional, and co. These are interesting words that some of us have actually heard at one point or the other. I'm not trying to make things difficult. I just want you to just follow till this point before I start to use bigger English to explain the concept for us. Sorry, do we follow me to this point?
Yes.
Okay, thank you so much for that feedback. All right, so I spoke about taxonomic tree. Now I will try as much as possible to point to okay, I don't think I can do that pointer. Okay, but I will try as much as possible to mention, to speak to the fact that not only taxonomic tree and our diversities can be getting through metagenomic sequencing, right? Our analysis. We can also, after assembling of the sequence, we can also start to find markers, gene predictions, and co. And these gene predictions actually serve different purposes. So for some gene predictions, it will help you discover antibiotic resistance. Some other gene prediction will help you discover some category of microorganisms that are your interest, urogenes and other kind of genes that are actually your interest, really. And there are a lot of functions you may also want to start to explore, like the cake function, gene function, the seed gene function, the COG seed function. We have amino acids. There are a lot of gene functions you may want to do if you are interested in intramolecular networking of what you actually have here. You are trying as much as possible to link it up with our normal body metabolism. I want you to understand that that you are able to do that with this. I want to revert to something. Now we're talking about sequencing. The truth is that after the sequencing read, immediately you already have your sequencing machine. Your sequencing machine will bring out something for you. It will bring out your data. Your data will be sent to you, but I need you to understand that from here downward, you need to know, you need to engage it with bioinformatics for you to make meaning. Again, do you follow me to this point?
Yes, please.
Yes.
Yeah, because everything you are going to get from your sequencer. I'm trying to reverse the slides. Everything you will get from your sequencers will look like this. You will not be able to make sense of it until you have until you have bioinformatics knowledge. Hence, your knowledge, hence you need to actually know. That's why this workshop actually comes in handy for you. So 16S ribosomal RNA gene sequencing gives us, provides us with the ability to describe complex microbial community that has been limited by the fact that many of these organisms, they remain unculturable. So in order to classify the microorganisms that make up the human microbe microbiomes, for example, wastewater body microbiomes, or maybe you're interested in stool microbiomes, or maybe gut microbiomes, I mean, skin, any of the environmental microbiomes. Scientists have used 16S gene sequencing, and the 16S rRNA is a component of the 30S subunit of prokaryotic ribosomes. I think we saw that in the last part. And the gene encoding the 16S rRNA is often used not only for the phylogenetic studies, like we can see here, of course, with the file, I mean, it's actually, I think I kind of explained this. It's not only used to identify bacteria. Using this, it can also help us to also what map relationship. By relationship, relationship, what crosses your mind when we talk of relationship? We'll be talking about their phylogenetic relationship. We're talking about their abundance relationship, their community relationship, their intra-community relationship, and any other relationship that we would love to draw out of it. So we can also even bring it to even the marker relationship, the gene marker relationship, and so on. So metagenomic sequencing is quite interesting because it provides us a good estimate of who is here in our sample. And this data do not only tell us, the scientists, about what microbes could be doing in our body. I mean, it could be present in our body and what they could be doing in our body. Also, it can also help us to understand, I mean, to look closer at our body in order to get a better idea of how these microorganisms, relate with each other. Scientists have actually turned to metagenomic sequencing for a lot of these things. So metagenomics is the study of the total DNA extracted from an environment. Instead of us to sequence it and do some of these normal, I mean, instead of us to culture it and do some of this our usual microbiology procedure, just get it and sequence it, and that is it. So I mentioned that previously, that what scientists actually do often is what they do shotgun sequencing from the sample. From the sample, when they get the sample, they extract the DNA, they cut the DNA, then they sequence it. Then that is actually one of the things that we actually see here. I need to also speak to the sharing, because I mentioned shotgun sequencing, and it's always it's crossing their mind. So okay, so the shot there means, let's say they cut it. So in shotgun sequencing, all the pieces of the DNA within a sample, they're cut into small pieces. The DNA has to be cut into small pieces because the machine that are used currently to sequence DNA are not yet good enough to sequence very long pieces of DNA. Of course, in recent times, we are having breakthroughs as regarding long sequencing. But I must tell you that the breakthroughs we are even having as regarding analyzing long sequencing, it's much more difficult for us to do the bioinformatics analysis of long reads than to do the bioinformatics analysis of short reads. They are short. It's much more easier for you to follow. But once they are long, you are going to actually spend a lot of time, and you need a lot of computing energy to do that. I'm speaking from a first, and from my own experience. So each of the small pieces is sequenced. So since the resulting sequence contains small regions that overlap with each other, with one another, computer programs can be used to piece them together. And and these sequences are are going to turn out as best as possible. So it's important to know that it is often not possible to create the entire genome of an organism present in a sample. Let me say that again. So it's what you are going to be getting after the whole sequencing. Let you understand that at one point or the other in your whole processing, you're going to maybe miss some one or two things out. Some one or two species out, or let's say contaminate some one or two species genome, for example. But what you are actually getting is actually better than, it's 90%, 98%, depending on how good you are, better than culturing, because in some culturing you can't even culture some of these things. So there are some interesting concepts we can actually see. I'm trying to zoom out so that we can see some of the things that we can get out of metagenomic sequencing. So some of these concepts are kind of peculiar, especially when you are using, you are doing an amplicon sequencing. We have something known as the marker-gene analysis. We have something known as the binning, assembling, and co. You get to see a lot of concept here. I remember somebody asked from me today, and the person said, so what stage did you get to with this person's work? And I said, well, after pre-processing, I've been able to get some BAM and some BAM file and some SAM file. And the person asked again and said, so can we really give this person this result? And I'm like, no. Some of those results are, comes with the concept of what is known as contige. Do you follow me? So I've just assembled them. I still need to go higher to align them so that later on, after alignment, I'm able to get a taxon. I'm able to get a taxonomic profile, taxon abundance profile, or something that some of us know as the OTU table, and the operational taxonomic unit table. And from there, I'm able to also do some phylogenetic analysis, and I can start to do other analysis. So let me go back again into shotgun sequencing, so that I'm trying as much as possible to make sure that the concept of shotgun sequencing do not weary us, because giving us a lot of details about it may actually be tiring for some persons. Again, scientists with shotgun sequencing, they are left with multiple fragments of different genomes. This fragment can be compared to a complete genomes of other known organisms, as well as can be compared using various databases of genetic information in order to figure out what bacteria were generally present in the samples, as well as what genes they have, that these bacteria actually have. And information about what genes are present helps us to predict what these microorganisms or these organisms are, what kind of function, what are the functions within the human body. I took time to explain this concept to you so that you can get it clearer. But at the same time, you're going to engage the platform, and the platform will bring you much details. From my own explanations, I tried as much as possible to simplify it. But now let us go and start to look at the technologies that we have in our time. So presently we have five different technologies, as it were. We have 6 different technologies as it were, because I can't find the other rochas and one other by Thermo Fisher. So I can't actually find them here. But here we can see a 3rd generation sequencing. a third generation sequencer. This is a second generation sequencer. This is a third generation sequencer. The first generation sequencing is known as Sanger sequencing. Okay. Everyone, did we get that? So the first generation sequencing is known as Sanger sequencing, what we know. The second generation sequencing is known as NGS, next generation sequencing. So anything after the second generation upward is called the next generation sequencing. So we have the Illumina sequencer here. We have the PacBio sequencer here. Illumina produces short reads. PacBio produces long reads and is able to do whole genome sequencing. Then we have the Oxford nanopore sequencing, and this is an example of their pocket size sequencer. Now, can you imagine this? Imagine that you are able to get this tool, and with the help of its workflow, sorry, its cell flow, easy next generation, third generation sequencing mechanism, you're able to get your sequence from an environment. This is quite convenient. Now look at this. You have to feed this one with power and run it for a long time. So there are different types of sequencers, like we can see here. We have the second generation, we have the third generation, we have the third generation Minion, third generation Illumina, second generation PacBio, third generation. Now, please, I want us to be penny wise here, and I think I should share at this point from my own experience. Every of these generations has their own advantages and disadvantages. I must actually say that you need to also confer with a bioinformatician so that they can advise you on what to do as regarding your own project. There is no sense going to choose a third generation sequencer because, okay, somebody just said that it is latest. Man, at the same time, you get stuck. You're not able to do your own analysis. When you are able to, when you have an option of a second generation sequencer that is able to also give you whatsoever you want, and is able to also give you something that, you are able to also follow the procedures very, very often. So, very, very well. So I'm actually saying this so that you understand that there is a place to consult with bioinformatician, analysts and consultants to provide you wisdom on what to choose. Don't just read it on a paper, because a paper says that, okay, you did this, we did that. Good. They are correct. They have the know-how. They consulted with somebody concerning you and your own environment. You should also consult. Yeah. Even though we want to replicate their result, we need to also be authentic. So from that, I go to speak to amplicon sequencing. 16S amplicon sequencing has been recently developed. It's a technique that is used for to study microbiome directly in its environment. So metagenomics sequencing that focuses on 16S, 18S ribosomal RNA that has to be amplified with PCR amplification. This is actually the concept of amplicon sequencing. So the 16S gene sequences actually contains something, these genes of interest in 16S. They actually contain a hypervariable region that is of interest to us. And that region actually provides species-specific signature. This is actually the technical way to explain everything I've actually said. And this species-specific signature is useful for bacterial identification. From that point, I move forward to the fact that bacterial 16S genes contains 9 hypervariable regions. Please, I want us to get ready for what is going to happen next. V1 to V9. So ranging from about 30 to 100 base pair long, that are involved in the secondary structure of the small ribosomal subunit. I think we can actually see this, but this is actually for the Escherichia coli, right? Uh, just a diagram, right? So the degree of conservation varies widely between hypervariable regions, which with more conserved regions relating to higher level of taxonomy, and the less conserved region helps us to be able to focus more on, to be able to get the genuses and the species. So the species level, we call it level 7, the genus level, level 6. I want us to know that uh species level is actually very, very okay. All right. Using species level to actually get the microbial community is quite very, very okay. But other times, a genus, getting the taxonomy getting the community of an organism, even at its genus level, is also very, very okay, because world researchers many a times they get stuck on the genus level and they're not able to come even down to the species level. But even as at that, they are able to establish relationship and bring forth meaningful things. So sometimes we are able to sequence even onto this onto the species level. Other times we are able to sequence onto the genus level, and there is nothing wrong in it. While the entire 16S sequence allows for comparison of all hypervariable regions, at approximately 16S, at approximately 1,500 base pair long, it can be prohibitively expensive to study, to try to study an organism by looking at all of its hypervariable regions. So it means that you have to start focusing on getting its V1, V2, V3. You actually have to start sequencing, focusing on different regions, adding primers and adapters that will help you to get the regions you are kind of interested in. So you need to be very, very, very penny wise to determine what hypervariable region, you understand, is strong for the kind of studies you are interested in. I hope you follow me to this point, so that you don't just go and choose a V9. You get stuck there. You get to discover that a V9 may not practically be able to give you what you want, because a lot of V9 hypervariable regions of 16S have not really, a lot of research have not been done. Maybe there are not enough database to help you to sort out your community of interest. Let me leave that there and continue. So I want us to understand that there are a lot of studies that have commonly utilized the Illumina platform that produces reads at the rate of 50-fold and 1,200-fold less expensive. And there's also another sequencer that I kind of mentioned that is not present there. One of the sequencers is the 454 pyrosequencers, and that is also not present there. The 454 pyrosequencer is very, very expensive. The Illumina platform, sequencing is relatively cheaper. The Sanger sequencing, I don't know, in recent time, I don't know if it's cheaper or much more expensive. I'm not too sure about the rate presently, but the Illumina sequencing has actually been quite interesting, especially in shotgun sequencing. Illumina sequencing only produces reads between 75 to 250 base pairs long, because it can produce up to 300 base pair long, especially when we use the Illumina MISEC. And it has no established protocol for reliable assembling process of the full gene in the community sample. So if you're interested in the full hypervariable region sequencing and how to assemble it from a single from a single Illumina run, you need to actually learn how to target these regions and do your studies. All right. So at this point, all right. So at this point, I know I'm taking a lot of time to explain, but let me just run to the end quickly, so that we can just look at this and get some. And just round up. We have something known as Silver. It's a comprehensive online resource for checking, for quality check, and to also get aligned ribosomal RNA sequence data. So Silver provides a comprehensive, quality checked, and regularly updated data set of aligned small, small 16S, 18S, RNA, and large subunits like 23S, 28S, ribosomal RNA sequence of all three domains: bacteria, archaebacteria, eukaryotic. The eukarya. We also have other online resources except from the Silver. We also have the green gene that, like you can see, 16S RNA gene database. You can also find RDP, and you can also find NCBI. So these are lovely database. We have OTT. I think I also found one there too in China. Then there's also another one I shared with a colleague. That one is actually 16S, but it's specific to viruses. I'm going to remember his name very soon, but it's quite interesting to see how that work. I've just shared about the database for the 16S, 18S, let me just speak to the general ribosomal RNA sequences, because you need to compare your sample with a reference, and you can find your references in the database. So let's provide a perspective to what we have been discussing. Here's an example of the American Gut Project, which has recently been moved to a new URL. The American Gut Project is the largest crowdsourced citizen science project till date. They got a lot of samples. I'm trying to zoom in so that we can see. Yeah. So over here we can see some sample types, from the fecal to the oral to the skin and other body sample site collected from thousands of participants that represented the largest human microbiome cohort in existence. So the detailed health, lifestyle, diet, detailed diet data among some other factors associated with the sample collected. We actually documented. This actually enabled us to deeply examine the association between human microbiomes, factors such as diet, from vegan to near carnivores to everything in between, to the season, to the amount of sleep, disease states such as inflammatory bowel disease, diabetics, diabetes, autism spectrum disorder. A lot of things was actually dug out from these samples. Yeah. So can you imagine how powerful your sample can be, especially even your gut sample? So again, the question comes again, that what is a microbiome? Microbiome is a collective genome of microbial population, including bacteria, fungi, viruses, bacteriophages, and protozoans that live inside and on the human body. So in an elderly adult, for example, the gut microbiomes exist in a state of mutual symbiosis with its host. So let me ask, what is a symbiotic relationship? Is any type of a close and long-term biological interaction between two different biological organisms, be it mutualistic, be it commensalist, be it parasitic. So the microbiota inhabiting the gut are estimated to contain upward of more than 1,000 microbial organisms. So the component of the gut microbiota can differ drastically between the regions of the GIT, that's the stomach, from the lumen, sorry, from I mean that's the throat, from the lumen to to the stomach, the lumen to the epithelium, to the mucous layer. I'm talking about the gastrointestinal tract there. So the important interactions between the host gut is microbial in inhabitants remain elusive due to much or such dramatic diversity, and this has actually marked differences. So there are a lot of distributions within the gut depending on where you are looking at. The sequencing of gut microbial genomes provides information useful for identification of pathogens, antibiotic resistant strain, and immunomodulatory microbial population. For some persons, immunomodulatory microbial populations is actually their own interest. They want to see, okay, how does what someone eat, how does it affect their well-being? How does it affect how they sleep? How does it affect their immune system? How does it affect, I mean, a lot of functions. And seriously, microbial gut microbiome has actually played a lot of roles even in such instances. Okay, at this point, I move to speak to the fact that the gut microbiome has an enormous impact on the life of the host, and the diet plays a fundamental role in shaping the microbiome composition and function. The way food is processed is a key factor that determines that determines the amount and the type of material reaching the gut bacteria, and it's also influencing the growth of this bacteria and the productions of the microbiota metabolite. In this perspective, the current possibility to address food design towards a better feeding gut microbiota are highlighted together with a summary of the most interesting microbial metabolites that can be made for dietary precursors. So we discovered that we are what we eat. So we are actually what we eat. If you eat good, if you're able to eat some particular things in which some particular things that will help the bacteria to produce the right metabolites for your body, you're going to see the result. They are producing the wrong metabolite for your body, you're also going to see the result. So so that's quite interesting. So at this point, before I go to the question section, I would love to mention that gut microbiota, they play a very vital role in the common types of diseases, right? The infant gut bacterial microbiome, they rapidly diversify over the first year of life, in an elderly infant. But it's kind of delayed in those that develops allergy. It's also delayed in those in children with asthma. It delays in children that are malnourished. So there is a number of of prenatal exposure, I will mention perinatal exposure, postnatal environmental exposure that are known to modulate the risk for childhood disease. And one of those things, those actually include the formula feeding. When you feed a child your formula food, some of these our local formula food, it actually has an effect on the child. Then when you use an antimicrobial an antimicrobial drug on a child, instead of finding other natural means to get him well, it's going to affect his gut microbiota. Also, exposure to environmental tobacco smoke, carbon exposure, and in fact, when an animal gets, when a child is exposed to animals, let's say your pet at home, and they're all around licking him, licking his face, you are giving your child a different exposure. So this same exposure, it relates to gut microbiome composition at discrete developmental time point. And it's very, very important to understand that whatever you expose the child to at the beginning determines what start to take base in his inside. So from developmental time to successional trajectory of his early life. At this point, I'd love to ask if we have questions.
Yes, I do. I have a question.
All right, please, over to you.
All right,
Okay.
sir.
First of all, I want to appreciate you for this wonderful session. It's quite informative and educating. I've really learned a lot. And my question goes in line with this topic. Though you mentioned it, but you didn't get to that point that is not really clear to me. And it's about targeting of genes. So I want to know if samples are collected from, say, environment now, and then we decide to run a metagenomic analysis. Of course,
Okay.
the metagenomic analysis will help us identify the organism and then probably tell us more about the genes contained in those organisms, maybe antibiotic resistant genes and the like. But I want to ask, I've been reading in journals, I've been seeing the V1 to V9, which you actually mentioned today, which has to do with various hypervariable regions in organisms, and you talked specifically on E. coli. But what if the organisms in that sample, what if E. coli is not part of the organisms in that sample? How then do we identify the genes to target or the genes to sequence?
All right. Thank you so much for that. So I would love to mention that the hypervariable region is present in every bacteria. All right. And by speaking of targeting one gene or the other, many a times we are not targeting the genes. Many a times we are just doing, we are trying as much as possible to get the total, to just get what we can find first. First of all. Then when we already have what we can find, then we can now start to focus more on what our own interest is. So let me say that again. So when you do a 16S, so it's from the beginning you determine, you ask from yourself, do I want to do a 16S? Do I want to do an 18XS sequencing, for example? So if you're actually interested in sequencing a bacterial, but you're not interested in the whole genome sequencing, but you are interested in, let's say you want to focus on okay, let me use that same example on finding bacteria that are resistant to a particular antibiotics, for example. In that sense, and in that case, you need to go and read, can 16S sequencing, can it focus on genes, or can it provide me variable regions of genes that helps me to identify antibacterial resistance gene? If no, what kind of sequencing do I need to do to get that? So at this point, I need to answer that question so that you don't go and start to look for it again in journals. So you don't do 16S sequencing if you're looking for bacteria's, I mean, for antibacterial resistance gene markers. So you do whole genome sequencing so that we are able to find some of these other genes. All right. So many times we do whole genome sequencing so that we don't miss out any regions, we don't miss out any other genes. Do you follow me to this point?
Yes, I'm following.
Yes. So you do so that you don't miss out that. So like I mentioned, we had the applicon sequencing, we had the 16S rRNA. Then the first one we met was what? Whole genome sequencing. So that was why I said that your own interest, you should consult with somebody, with a bioinformatician that will be able to tell you that you can't use 16S sequencing to get this. So many of the mistakes that please, Dr. Adekunle, please get ready. So many of the mistakes that our own researchers here in Africa that they do often is that they will first go and do their own sequencing. Then they'll come back to meet you as a bioinformatician that you are the miracle worker. You are the miracle worker. But we can't bring out, we can't just bring out results from nowhere. Any result that is not, any analysis or any pathway that the kind of sequencing you do limits the kind of result we can get. So if you want to get result on microbial population, you face a metagenomics analysis, and one of the analysis will also, will actually involve you doing a 16S or a whole genome sequencing. Note, whole genome sequences are much more expensive than 16S because a lot of persons are doing 16S. It makes it a little much more cheaper. So I don't know, have I answered your question?
Yes, to an extent, but I'll consult you more later.
All right. Yeah, I think I can see Tauma in my hand. Please, your hands, please. Please, you can speak. I can't hear you, Ima Taona from Zimbabwe. Are you there? Please, can you speak? Okay. I don't know if we muted, but we can't hear you. Ima Taona, we can't hear you. All right, so let me call on the next person. I saw the hands up. I think I saw Dr. Popola's hands up.
All right, thank you, sir.
Yeah,
thank
welcome.
you for this section. It's really been an eye-opening section. Sir, my question is, I actually have worked on metagenomics, even though I didn't really understand what I was doing. I just collected sample and I think I used shotgun. I use shotgun sequencing. So now I think I have an understanding now that I cannot target, if I want to target a particular area of interest, I cannot use 16S. I have to use, I should use 16S instead of shotgun. I should use 16S according to what I've heard so far. But now, looking at my question now is, I want to really get the basis and understanding. 16S, what is actually the difference? If, for instance, I want to target, I have something I actually want to target, what is actually the difference between the 16S RNA and the 18S? Is there, I didn't get when you were talking about the ribosomal. I was distracted. But I really want to, is 16S, most of the time I hear people use 16S. I hardly hear people use 18S.
Okay.
So, so that's
All
actually my
right. So let me mention that again. So shotgun sequencing. I'm going to share my screen again so that you can see it. I'm sorry, Dr. Adekunle, let me just make this clarification so that you can take over. Quickly. So listen, there is nothing special in shotgun sequencing. Okay. What does shotgun sequencing bring us? Sorry. I'm not too sure. Can you see my screen?
No, we saw it, but it's not there again.
Okay. Sorry, let me do that again so that you can see it. Okay, I think you can see it now. So shotgun just speaks to the fact that you pieces it. I mean, you pieces your DNA. Okay.
All right, sir.
It's just a way to just describe the process. It's not really like I mean, like shotgun. And whenever you hear shotgun, it should just be like short reads. I mean, like, okay, I will get short reads. Okay. The real question I believe you are asking for me has to do with this. 16S has its own limit to what it can target. Amplicon sequencing has its own limit to what it can target, has its disadvantage and advantage. Whole genome sequencing has its advantage and disadvantage. So for all of them, depending on the kind of sequencers you use, most times 16S and amplicons are able to do shotgun sequencing. I mean, it means that after you have extracted your DNA, you can cut it into pieces. Then you can now put it inside the sequencer and it can run. In the long run, it brings out short reads for you. You understand me? But the whole genome sequencing, it has its own protocol too. And it also has what it can also target. So whatever you want to target, we need to actually understand it from the beginning so that we tell you that don't go and do 16S or go and do amplicon sequencing. With amplicon sequencing, for example, you go and do PacBio, for example, and when you do PacBio, okay, you should be able to have this kind of read. Then we are able to find what kind of marker you're actually looking for, and so on. So you should not have finished doing the sequencing. Then in the end, we are just trying to salvage what we get, and what we get is not actually your own research interest. I mean, it's not actually. I mean, it doesn't speak to the fact that the project was planned, especially when we are trying to salvage result from a process that I mean, after sequencing have been made. At the end, you are not satisfied with your project because you are not able to get the marker that you are interested in. Please, did I answer your question?
Yes, what of 18S?
So speaking to 18S, many times we use 18S to find fungi. So they are very, very strong in bringing us, I mean, in helping us to identify fungi. But at the same time, there are some bacteria that we can use. We can use 18S to find. So towards the end of my presentation, I spoke of some databases. You can observe that some of the databases, for example, the green gene is strong with what? With 16S.
Yes.
Yeah. Whereas the silver is able to produce you 16S. I mean, give you 16S and 18S small subunit RNA. Sorry, I mean sequence. I mean reference sequence, and we also have 23 and 28. So for the mycologist and co, we are interested in only fungi. It may even be fungi of the foot. it's quite advisable for you to run 18S. So so that I believe that I am able to answer your questions. Am I able to do that?
Thank you, sir. Yes, sir.
Yeah, please, I would advise that we
I
read
will
this,
add to that.
research paper that we can see on the screen. Yes, sir. Over to you, sir.
Okay, I think Anju also want to speak. So, Dr. Popola, I would
Yes,
just say in
sir.
a simple way, the main difference between the 16S RNA sequencer and shotgun sequencer, just as he has said, is the fact that for 16S, it targets and reads specific region of the 16S rRNA, and these are found in all bacteria and archaea. All right.
Sir.
Whereas shotgun, sequencing reads all genomic DNA in that sample, and it can include both bacteria, fungi, viruses, and other types of microorganisms. So if what you have done rather, was to go for shotgun sequencing, it means from your reads, there are reads for viruses there. It is not
Yes.
as
Yes.
target compared to when you do 16S rRNA. So if, for example, I'm trying to conduct a study that has to do with maybe looking at fungi in a sample, just like he mentioned earlier on, or viruses, then I wouldn't go for 16S rRNA because apparently I would have missed out both fungi and viruses. Okay. So,
Yes.
whereas, of course, the singular difference, if I would say, from 16S and 18S, 16S is something I would want to target if I'm dealing with prokaryotes. Whereas if I'm dealing with eukaryotes, then I will have to go for 18S. So that is why what I'm interested in doing, my project work or area of focus, to a large extent also determines what method I will want to use. 16 RNA sequencing is used to identify bacteria and archaea, like I said, while 18S sequencing is used to identify and compare eukaryotes such as fungi, protists, and even animals. Okay.
Yes.
And their base pair, is also varies. For 16S, we said, of course, there are 9 hypervariable regions, and there are regions of interest to 16S, amplicon sequencing and the rest. Okay, especially V3 to V5 and thereabout. Whereas 18S also have some other hypervariable regions of interest. So these are some common differences between them, but the basic or the most, I would say, main difference would be the fact that what if it is protists, animals. I'm aware of a project. I'm not sure if the person is also on this call, whose focus of sequencing were worms. I mean, and the rest, within the human, ecosystem. So apparently, what would be targeted in that kind of study would be an 18S sequencing, not 16S, because
Yes.
if we focus on 16S, we won't be seeing, what we are trying to target here. We only have access to archaea and bacteria. So I think those are just some of the basic differences. Whereas, of course, the method also differs. Is it shotgun or 16S I want to go for? Shotgun is a bit wider, but not as wide as the whole genome sequencing, metagenomic sequencing. I think those are just few things I would like to add before. But I think Andrew's hand is up. Um,
Uh, before Anju speaks, I think Ima is having an issue with her sound, like she mentioned, but she wrote a question in the chat. So Anju, please stay tuned. Let me just read Ima's question in the chat. She said, I have problem with my sound. Okay, so after whole genome sequencing of all the microorganisms in the sample, including bacteria, fungi, viruses, how do you differentiate if you are focusing on bacterial only? That's a very, very lovely question. So let me clarify something here now. So you can also use 16S to get viruses, because viruses too, we have our RNA viruses. Do you understand me? So it's quite interesting how that we can use 16S sequencing and metagenomic analysis to also do some viral studies first hand. But how do we differentiate them? So when you carry, when you have your sequencing, your sequences, and you plot it, or you place it, or you align it against a particular database or a reference database, what we are trying to do is that you are sieving it through it. So if it does not fit into the bacteria, for example. So in the process of bioinformatics, we have some phrases that speaks to the fact that, okay, except remove archibacteria, I'm not interested in it. Remove fungi, I'm not interested in it. Remove viruses, I'm not interested in it. I'm only interested in only bacteria. Then and there, so this stage actually after, comes immediately after you already have your data. It also comes immediately after you have denoised your data, after you have removed some of these other extraneous things. I believe that in part 2 you should be able to get more details of this. So after you have denoised, after you have pre-processed, and you have done a lot of these things, you will get to this point where you need to do the sieving. So there is a word we use for that. So then you plot it against this reference relatively. So you place it against the reference on alignment and co. You would be able to only get the bacteria only, the one that you are interested in. After the result you will get from this process will determine your OTUs or your ASVs. So OTU is your operational taxonomic unit. Your ASV is your amplicon something variant. So in short, they are the same. So when you already have that, you're going to have that table. So I believe I've answered your question. Have I? So the amplicon sequence variant really are quite interesting. Quite interesting to explore. I believe I've answered your question. Okay, yeah, you say yes, thank you. All right, Anju's hand was up. Is she still here? Anju, are you here?
Yeah, sir. Hi.
Yeah, hello. Your question, please. Over to you.
Yeah. First of all, your presentation was very nice. Can I ask my doubt? You show one slide about this prokaryotic and eukaryotic ribosome subunits, right? So in that slides, you mentioned 16S rRNA as 21 proteins. So I need to know, while doing sequencing, they are counting these proteins to identify the 16S rRNA region.
You are
Hello, sir.
muted though, we can't hear you. You are muted.
Sorry. Yeah, sorry about that. So I said that your question is when we are sequencing, how do we focus on 16S sequencing? Is that your question, Anju, right?
Yeah. Is there any connection with these proteins? Actually, that is my question.
Okay, so is there any connection between the proteins of the 16S sequencing, right?
Yeah. 16S rRNA proteins. And in the slide, you mentioned about 21 proteins, 34 proteins like subunit.
Okay. Now I understand. Yeah. So
yeah.
I think you're talking about this slide, right?
Yes.
So again, I want you to understand that the concept of the ribosome is something we used to know, and we understand that there is a binding site for this large unit and this small unit. So these units are quite stable. Okay, they are quite stable, and there is a gene factor to why we focus on this unit. So the previous speaker spoke about the fact that that we have, that why don't we choose 18S? That we choose, that why do we always focus on 16S, 18S? All right. Now I want to answer the question here, because your own interests determine what you do. The process of sequencing from the beginning is going to be the same for 16S, for 18S. But at the same time, to make sure that you have the optimal results, you make sure that your adapters and your primers are focusing on you getting the best 16S rRNA sequencing. Let me say that again. So from the very, very beginning, if you know you want to do a prokaryotic 16S rRNA metagenomics process, you should know that from the very, very beginning of sequencing, you will need to from this very, very, very beginning, you need to make sure that you are treating the whole process. You are following the right process, and the right process will include you adding an adapter, you adding a primer, and these primers and adapters should be able to amplify your 16S characteristics. So in your wet lab, do you understand me? Then that is not the only way you need to ensure that your 16S result is amplified. Also, after processing, after pre-processing, after you've imported your data into your bioinformatics field and so on, you need to denoise. Yeah. So you need to denoise, and you need to do some of these other procedures. Then relating to the previous question, when the person was asking about 16S, when you are aligning to your database, you should also make sure that you're selecting the fact that, okay, the 16S of my own interest is bacteria, and not the 16S of viruses, and not the 16S of protist. Did I answer your question, Andrew?
Some part I understood, but I need to ask one question: that is, this 21 proteins play an important role in sequencing or not? 16S rRNA region I understood.
So you want to know if the 21 are, if the 23 or the 21 S, if they play a role in sequencing.
No, if I can come in here, please. The 16S rRNA, essentially what this is saying is that they translate or produce about 21 proteins from,
I think we are missing you. We are missing you.
Translation. The can you hear me now?
Yes.
Okay,
so
the 16S rRNA, the ribosome RNA, produces about 21 proteins. We know from the molecular, the dogma of molecular biology that, of course, from transcription to translation and ultimately production of protein. So yes, our ribosome RNA proteins, ribosome RNA produces proteins, and we are saying that about 21 proteins are produced by this 16S subunit. Okay, so apparently, yes, they play a role in whatever downstream that we see. And of course, the proteins we'll be seeing, we have, relations to those specific bacteria or archaea that we are trying to identify or classify. Okay, so 21 proteins are produced from this 30S subunit. I don't know if that makes some sense. So that makes some sense. Andrew, did you get that part? Talking about the relationship between the
Yeah,
proteins
so
that are produced from those 16 sRNA subunits.
yes, sir. So can I ask one more thing?
Yes, you can ask. Perhaps if we can answer here, we'll take it up on the group chat.
Yeah, so this 21 proteins will play a role in this if peptides. In the case of if some bacteria will have some antimicrobial peptides also, right? So by identifying these like proteins, these are forming peptides or not? The same 16
Okay, I
sRNA
think I
protein.
understand your questions. I think I understand your question. So metagenomics analysis don't focus on proteins. Metatranscriptomics analysis focuses on proteins. What we focus on is the gene that produces these proteins. So the gene that produces this protein is what we tagged as 16S rRNA gene. Do you understand me? So
Yes.
our concern is not the protein. Do you understand me? So that was why at the very beginning, I mentioned that there are 4 different ways in which you can decide to study microbial community. Okay, and this is what I put here. If you want to study proteins and co, you need to go to metatranscriptomics. Here you are going to follow another process, but you have to start to deal with enzymes. All right, there's no problem with this. But over here, what we will focus on is gene sequencing. And one of the gene sequencing that is of interest to us is the 16S rRNA gene. Okay? So you don't need to worry yourself about worry yourself about other genes. All right, over to you, Dr. Adekunle, sir. I hope I answered your question, Dr. Adekunle. I mean, Andrew, did I answer your question?
Yeah.
All right. Thanks so much. Dr. Adekunle, over to you, sir.
All right. Let me know if you can see my screen. Can you see my screen, please?
Yes, please, we can.
All right, thank you very much. Once again, I want to appreciate every one of us. I will try to run quickly through the remaining sections so that we can end in the next 5 minutes or so. During the course of the next 8 days, we will be going through this program. And like I shared earlier on, we have 3 courses that are part of this session besides today's live session, which we would be sharing with us, all the participants on the Metagenomics group page, shortly. And equally, we look forward to every one of us engaging with these courses. Like I said, the first course, Metagenomics, has 13 lessons spanning an average of 25 hours. And we equally have the role of high-fat diet on obesity. This is a project that is meant to drive home some of the things we've learned. And equally, we have effect of dietary fiber intake on microbiome. Of course, these intakes, their environmental, factors. I'll put them as which plays an impact on the microbiome that in turn plays an impact on the outcome we have, functional outcomes we have in the system. So let's remember that this training is available and is accessible, for a variety of persons and people of all backgrounds, scientific in the scientific research space, students, undergraduates, postgraduates, or even PhD and faculties, as well as biologists and clinicians. So we look forward to having a very rich engagement during the course of this training. And I'd like to mention, of course, that our approach in Pine Biotech is an omicsology is to use a blended method of live sessions like what we have today, access to asynchronous courses, meaning self-paced, individuals beginning to access them at their own pace individually, and then also providing access to some of our platforms such as the T-Bioinfo platform, which is a cloud-based bioinformatics analysis platform, as well as some code-based too. You will have noticed from the content shared that some of those courses are code-based. All right, so that is very important for us to note. And of course, this here on the platform is the how the TBA info platform looks like. All right. Please, during the course of this program, reach out to us. Of course, under each of those sessions where you see a drop down button like this, there are demo pipelines that you could use. But depending on the course you are taking and what needs to be done, if you need to also have access to this, an educational access or students access, I will call it, okay, meaning you can bring in database to run, or sorry, data sets to run on these platform. But of course, there are those you could that are part of this project that you could equally lay your hands on to run by yourself on this platform. So if you have any access issues, kindly let us know. But of course, we have different parts to these platform. All right. So moving on. Unfortunately, many of our participants has dropped. I mean, participants in today's session. But moving on, it is my hope that every one of us here have our registrations done already. So all you need to do, like we said previously in some of our past workshops, that being able to register on this platform gives you access is a lifetime access, just as your Gmail account. I mean, it's available to you any other time. You can always log in to access any of our courses, or be part of our trainings. So for those who have been part of our previous training, you don't need to register afresh. Of course, all you will need to do is to log in and then gain access to the courses, which will be providing the specific URL for those courses to us as we move on. Okay, but I like to mention, let's not forget that there is a need for activity tracker, which has been shared via email with us. So we need to complete the activity tracker so as to, as we move on with this particular program. And we expect us to
Hello,
indicate the
sir.
startup once. Hello, can you hear me?
Sir,
We
I have
expect
a
us to
doubt.
indicate. Yes, so please, we expect us to engage
Uh,
with this activity tracker. All
I'm
right.
sorry, Andrew. You're going to ask your question after he's done, okay?
Yeah.
Andrew, okay?
Yes, yes. Okay,
Okay,
yes. Oh,
okay.
sorry. I didn't even hear you. All right. So, but I like to mention, don't forget that we have certificate of completion for each of those courses. There are 3 courses, included in this training, and we expect all participants to get all the 3 courses. Don't forget, from one section when you're done getting, interacting or engaging with the content, um, endeavor to click complete and then move next. Don't just click on the next. Follow those processes. When you click complete before clicking next, it's a kind of, milestone measurement to show that, oh, you have gone through this, this part of the course and moving up to the next. Ultimately, at the end, it gets to 100%, and then your certificate is automatically available. But beyond that, there's also going to be a workshop certificate we'll be giving at the end of the training. And let's not forget, that the part 2 of this course will be available also starting from 30th. So this particular one runs from 16th today to 23rd next Saturday. So there will be a gap in between. And by 30th, again, it's going to be a Saturday of September. We'll run the part 2, from 30th September to 7th of October. Of course, there we'll be looking at gut microbiome dysbiosis and its effect on non-antibiotics and Rett syndrome. We'll look at skin microbiome in psoriasis. And last but not the least is a bioinformatics project course that tells you how to design your own project and how to have access to different database and a whole lot of other stuff like that. Importantly is the fact that for those who have registered and paid the discounted amount for this course, let me mention that the cost for this the current part 1 bioinformatics training is $20. However, the original cost, if you go to our platform, is $45. So based on some scholarship and discounts availed, I think about 56% is off for individuals to pay 18,000 naira rather than $45. Now, beyond that, by the time we come to part 2, for those who have completed part 1 and actually did complete the courses, so it's not just that I've registered, I paid back then. No, we want, because we want it to be kind of a pedagogical learning, particularly for those who are part of the part 1. So if you have done that, rather than paying another 18,000 naira for the part 2, there's further discounts that participants only pay just 6,000 naira. I mean, to just take part in that part 2. But part 2 will equally be open for just anyone, newbies and anybody to be a part of it. Of course, our courses are recursive, meaning around the circle. I mean, at any point you join, you can always go back to also still take the previous one afterwards when being organized next time. So that is why outsiders or first timers can equally participate in part 2. And but of course, they will be paying a full amount. On that note, I think we have come to the end of this section. Other questions we can take on the group page and regarding registration payment and all of that. Let me state that please, after now we'll be engaging with those who have paid, because on the WhatsApp group page we have about 15 or 16 or more persons, okay, who are part of that group, but not everybody has fully registered. They've not made their payment. So it's only those who have made payment. We've already forwarded their details across to the international office, and activation of their accounts will be done so that they can have full access to those courses. For other persons, all of the courses, we'll share the link there. But I'm not sure you can access beyond lesson 1 of each of those courses. Take, for example, Metagenomics that has 13 lessons. From after lesson 2 downwards, you won't have access because they are premium courses, they are locked. But for those who have paid and their activation has been done, they will have free access to all of the lessons, which is the focus of this workshop. So I think further engagement will take on our group page, and we will be available to guide us through and go through this journey together the next 1 week. On that note, I want to say thank you very much. Please, Andrew, your question. I think Dr. Anfe is still handy also to take it up from there.
Yeah.
Thank you.
Thank you, sir. Sir, shall I share my screen? I attended the introduction of bioinformatics. After that, I can't able to click on the complete lesson like that to move to next thing.
Okay, please share.
What I have to. Sir, is it visible?
No, it's not visible yet.
No, it's not. Okay, 1 minute. Sir, now? Sir, now it is visible?
Yes, we can see your screen now.
So I can't click this complete button to move to next session. How? how to move forward.
Okay. I understand that this is introduction to bioinformatics. All right. Let me write you on Telegram, sorry, on WhatsApp, or another of our admin is going to write you on WhatsApp, so that we are able to see how to really help you with this. But wait. Uh, your lesson one showed completed, so I think you completed it. Can you see?
So yeah, so I have to click on lesson 2 like that. I have to move, right?
Yes. You need to go like from lesson 1 to lesson 2 to lesson 3. Yeah. So
So
you will not see next. So you need
okay,
to go to
so
lesson 2.
I
Yeah. Continue like this. Yes.
So yes, so when no move up, please. It won't work that way. Move up a bit. So when you have engaged with this content, scroll down a little, please.
sir.
A little more, please. Go down.
Yeah.
Yes, stop there, please. Stop there. So you see complete. You have to click complete. Kindly do that. Assuming you have
It is
engaged with the content.
no.
Yes, you must have done that before. So after then you click next. It is when you click next before you
Yeah.
go to gene prediction. Otherwise, gene prediction will not
Yeah.
open.
So
So,
I have a
but in some
session
instances,
for all these lessons.
you said what?
I have access for all these lessons, right?
Yes, I think this is part of the previous
Yeah.
workshop. You have access to all of these. But if you should, we've not shared the link for the first course, the metagenomics course with you. We'll be dropping that link as soon as we have information that the activation has been done in a short while from now. So once we share that, but you are sharing your screen, right?
Yeah.
Kindly let me share mine.
Yes.
Okay, so let me know when you can see my screen, please.
Yes, sir.
Okay, so I'm trying to open that page now. Sorry, it's taking a bit of time. Okay, so let me go back to courses. All right, so let me just we'll share the link to this course. So this is the first course we'll be engaging with in this program. If you noticed, you'll start from here. This lesson 1, Introduction to Metagenomics, a kind of overview again, yet again. And most of what we talked about today, you get to see that. Then we'll move on to the lesson 2, the microbiome and human health. After that, you notice metagenomic study and importance. There's a star here. For those who have not paid and who do not have their accounts activated, cannot access this course. Let me try and show you that. When you click on that, what you get to see will be, directing you back. Can you see that? It won't open. So only paid participant whose account has been activated, we have access to this and this and this and this and a whole lot of that. Please, when you get there, you can get here, you can click on load more to see other courses. Up until lesson 13, they are all starred. Do you get that now? Is that clear,
Yeah.
please? Yes, so please. But definitely, like I said, your account will be activated shortly because, of course, we've received your registration and payment, and you can take it up from there. I hope that is fine.
So
I hope that
after you
addresses your
sharing the
question.
link, we will.
We will be dropping the link on the WhatsApp page, the group page for this course.
Okay, sir.
Yes. Thank you. Dr. If over to you, can please wrap up. I will have to also leave.
All right. Thank you so much, everybody, for joining us today. Thank you so much for your patience. Mrs. Babalola, it's so lovely to have you here. Dr. Fajiri, lovely to have you here. Dr. Popola, so lovely to have you here. Andrew, it's so lovely to also have you here. It's a pleasure to have engaged us today. Thank you so much for joining our section. I believe that a hands-on practical is going to come on very, very soon. And this hands-on practical will be something I will want you to engage. So we look forward to having you join the program and to also work on your computer. Before the hands-on practical, I believe we will have you, we would have sent you some instructions of what you should do because you may have to work on Google Colab so that and try to do try to learn some programming about Python. Yeah, so thanks so much, everybody, and have a great evening. Cheers.
Thank you, sir.
Thank you.
Thank you, sir. Well done, sir.
Yeah,
Thank
God
you, sir.
bless. Yeah, thank you.
You too.